Students, welcome back to Recombinant DNA technology, and in this session we are going to talk about the different basic steps which have been present in recombinant DNA technology. So the first step that which we need in genetic engineering or recombinant DNA technology is to isolate the vector as well as the target gene. You know what is a vector? A vector is something which will carry your gene of interest, and your target gene is something which will code for your protein of interest or the substance which you need to produce. And the second step involves cleaving or cutting both the vector as well as the target gene using the same restriction enzyme. The third step involves the joining or ligation of these cut vector as well as the target gene. The fourth one involves the transformation or to put the recombinant DNA onto a host cell. The fifth step involves cloning it, and sixth one mainly includes the screening of the recombinants. So let's just go through the steps of these in detail in a brief manner. So the first step is the isolation and the purification of the DNA. Both the vector as well as the target DNA molecule can be prepared by a variety of routine methods, which are not discussed here. So you can, in either cases now, if you have to take a gene of interest from a particular human DNA, you have to isolate the human DNA. And if you want to put it into a particular vector, let it be a different vector. I told you a vector is something which will carry the gene of interest. So you have to take the vector DNA also. Both of these DNA, let it be the vector DNA and the target DNA, that have to be isolated by routine methods. You know about the DNA isolation methods which we have come across by various techniques in the, like there are standard protocols for that. You can either use a kit or you can also use standard techniques to isolate the DNA. I'm not going into the details of that because otherwise we'll be going a bit longer. So once you isolate your DNA of interest as well as the vector DNA, you need to cut both of these DNA by using restriction endonucleases. The cleaving of the DNA to generate the fragments of defined length or with specific endpoints is very crucial in recombinant DNA technology. If you have to take a gene of interest from human DNA, you need to take only that particular part of that DNA. You do not need to take the whole DNA and put it into a bacteria because that is not possible. Only the gene of interest or the region of interest which you need to combine with a vector is needed for the recombinant DNA technology. So once you isolate the human DNA, if it is a human target, okay, you splice it and you get the insert DNA. And at the same time, you also have the vector, and the vector also has to be cut using the same restriction endonucleases as we had mentioned in the previous class. And once this restriction digested vector DNA as well as the gene of interest has been isolated, you need to ligate it so that it forms a what? It forms a recombinant DNA. And that is usually being done with the help of certain enzymes called the DNA ligases. These are commercially produced from T4 DNA ligases. You can purchase it from a supplier, and you can use this ligase enzyme to ligate the DNA of interest along with the what? Vector DNA, so that a recombinant DNA or a chimeric DNA is being formed. Now, after the process of ligation, you have to introduce this recombinant DNA into a compatible host cell. What is a compatible host cell? A compatible host cell is a host cell which will receive a recombinant DNA. Okay, and which will allow the recombinant DNA to replicate within it. In order to be propagated, the recombinant DNA molecule must be introduced into a compatible host cell where this can replicate. And this introduction of recombinant DNA into the compatible host can be done by various methods. Either you can do by genetic transformation techniques, or sometimes you can also use these can be packaged into viruses particles and then transferred into the host cells by transfection and all that. The various other techniques which are being used we will be discussing in detail later. So the fourth step mainly involves the transferring of the recombinant DNA into the compatible host cell. And once it has been transferred into the compatible host cell, within the host cell, the cloning vectors, they will, these recombinant DNA will be what? It will replicate. And sometimes the expression of the recombinant DNA also will happen in the host cell. The cloning vectors will allow insert DNA to be replicated. And in some cases, you can see that these cloned vectors will be also being expressed in the host cell. And the ability to clone and express the DNA efficiently depends upon the choice of the appropriate vector and the host cells. So, see, sometimes you just need to what? You just need to introduce a particular DNA into the host cell and you need to replicate it. So in some cases also you need to express that particular DNA in a very high level. So depending upon that, if your purpose of transferring the DNA is just cloning, you have cloning vectors for that. And if you want to express that particular protein in the host cell at a very high amount, you have expression vectors for that. So depending upon what is your purpose, it will change. Now, identification of the host cells that contains the recombinant DNA of interest, that is also an important step, which is it, and it is also one of the most difficult steps in recombinant DNA technology. The vectors usually contain certain markers, like now for example, they will contain genetic markers or genes which will allow the selection of host cells that have taken up the foreign DNA. Now, it could be a character like ampicillin resistance or something like that. Or it could be a oxotropic mode or something like that. Oxotropic type of nutrition which was being coded only by the vector. So, different characters, usually ampicillin resistance or antibiotic resistance is the most commonly used genetic marker. So, if the vector is being entered into the host cell, what happens? The host cells would become sometimes antibiotic resistant, or sometimes it would be undergoing a color change. That would be used to identify whether the host cell is got the vector DNA which is already combined with the with a gene of interest. So the identification of a particular fragment, it usually involves an additional step, that is a screening of a large number of recombinant DNA clones will be done, and you will identify which host cell has got the recombinant DNA. So if you go to consider genetic engineering, it mainly includes 6 main steps as we had mentioned earlier. And these steps includes isolating the vector as well as the target DNA, cutting it with restriction endonucleases, joining it by the process of ligation, transforming it into the transforming what? The recombinant DNA into the host cell where it can multiply rapidly, then cloning, that is the host cells are being what? The host cells are being cloned or multiple copies of the host cells are being created, and then you screen the host cells to see whether they are having the recombinant DNA or not. Okay, this screening can be done by various methods depending upon what type of vector you are used and what is the marker which has been present or which has been associated with that particular vector. So we will be going to the details of this in the coming sessions. Thank you for now.